Hello guys, I am Shubham from Tutorials. In the previous video हमने देखा स्प्लिसिंग। ओके, प्रोसेसिंग हमने कंप्लीट किया। अभी स्प्लिसिंग का ही और एक टाइप है, अल्टरनेटिव स्प्लिसिंग, which we will be discussing in this video. So before we start, subscribe to the channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. Alternative splicing is a type of splicing. ओके, स्प्लिसिंग का ही प्रोसेस है, लेकिन इसमें क्या होता है, I will draw your a mRNA. Okay. First of all, I will draw with different colors. Okay. Okay. So this is the mRNA. 5 prime end है and 3 prime end. Okay. ये different colors में मैंने जो निकाला है वो है exons. Okay. So I will write here E1, E2. This is red. Orange वाला E3, pink वाला E4, and again green, which is a E5. Okay. So this is a exon. Right? Exon and intron. जो black है वो intron है. Intron 1, intron 2, intron 3 and intron 4. राइट? हमने अभी क्या पढ़ा? रिमूवल ऑफ इंट्रॉन्स। ओके, इंट्रॉन्स को रिमूव करना स्प्लिसिंग है। ओके, बट इन दिस अल्टरनेटिव फॉर्म ये प्री एमआरएनए है। आई विल राइट हियर प्री एमआरएनए। प्री एमआरएनए से मैच्योर एमआरएनए बनेगा, राइट? व्हिच विल बी ऑफ टू टाइप्स। ओके, टू टाइप्स कैसे? लेट्स सी। इन द फर्स्ट टाइप आई विल जस्ट ड्रॉ फर्स्ट। So in this first type of mRNA, E1, okay, this E1, E2, E3, E4 are retained. Okay. Now in the second type में क्या होगा? Second type में E2, E3, E4 and E5. Okay, this is a 5 prime end, this is a 3 prime end. Okay, so these two types, मतलब एक ही प्री एमआरएनए से हमें दो प्रकार के एमआरएनए मिले, राइट? In the first case, we have exon 1, exon 2, exon 3, exon 4. Okay, this case में different protein. In the second type, we have E2, E3, E4 and E5. First case में E5 missing है, and in the second case E1 is missing, right? So these are the two different types of mRNA which will code for obviously different proteins, right? जो proteins बनेंगे उसका structure कुछ अलग-अलग होगा, okay? But these are formed from this single pre-mRNA. Okay, so this process is known as alternative splicing. Okay, अभी हम पिक्चर से देखेंगे और थोड़ा एंड विद द एग्जांपल्स एक्चुअली। तो इसमें एग्जांपल्स भी बहुत इंपॉर्टेंट है। दिस इज द एग्जांपल नाउ, अल्फा ट्रोपोनिन एंड बीटा ट्रोपोनिन टी, वेरी इंपॉर्टेंट। अभी यहाँ पे क्या है, प्राइमरी आरएनए ट्रांसक्रिप्ट। इसमें एक्सॉन 1 है, एक्सॉन 2 है, ओके, 3, 4, 5 एज आई शोड। ओके, 5 एक्सॉन्स है यहाँ पे, बीच में इंट्रॉन्स है। अल्फा ट्रोपोनिन में क्या है, 1 है, 2 है, 3 है एंड 5 है। 4 is missing, no 4, right? Beta troponin 1, 2, 4 and 5, no 3. Okay, मतलब in alpha में 4 missing है and beta में 3rd missing है. Okay, got it? So इससे two different proteins, alpha troponin T and beta troponin T. So this is the example, right? Got it? मैंने भी पिक्चर से अभी वही समझाया। 5 एक्सॉन्स, एक एग्जांपल में 4 एक्सॉन्स आए, 1 2 3 4, इन द सेकंड केस 2 3 4 5। गॉट इट? सो यहाँ पे और वेरिएशंस क्या-क्या आ सकते हैं स्प्लिसिंग में? तो देयर आर डिफरेंट टाइप्स एक्चुअली। सी, फर्स्ट ऑफ ऑल ये डीएनए है। एक्सॉन, इंट्रॉन, एक्सॉन। You can see here. After transcription RNA, primary RNA transcript, pre-mRNA we can say, right? Pre-mRNA. अभी इससे क्या-क्या होगा? In the first case में हमें क्या दिख रहा है? Just splicing, just splicing. Okay, this is the exon 1, exon 2, exon 3, normal splicing. तीनों भी यहाँ पे है, so this is normal. Okay, splice mRNA. अभी in the second step exon skip. Okay, exon skipping ये भी होता है. We can call this process as exon skipping. हाँ, क्या हुआ यहाँ पे 1 and 3? Okay, only 1 and 3 is written, 2 is not there. Okay, 2 को skip किया. Which exon is skipped here? Second one. In the mRNA, spliced mRNA, only exon 1 and exon 3 are retained. In the third step में क्या है? Exon extended. Exon extended मतलब यहाँ पे जो exon है इसके साथ इंट्रॉन भी आ रहा है. See, small part of the intron is retained. Got it? Intron is retained. You can see here from the diagram. This much part of intron is retained. Right? अभी फोर्थ स्टेप इंट्रॉन रिटेन मतलब कंप्लीट इंट्रॉन. ये कंप्लीट इंट्रॉन इज रिटेन हियर. फुल इंट्रॉन. यहाँ पे इन द थर्ड स्टेप थोड़ा सा पार्ट था इंट्रॉन का. इन द फोर्थ स्टेप पूरा इंट्रॉन रिटेन हुआ है. राइट? एंड इन द लास्ट स्टेप अल्टरनेटिव एक्सॉन्स. Alternative skipping, splicing. Okay, see, two products are there. This is the first, this is the second. यहाँ पे 1 and 2 है, which will code for a different protein and different type of protein we can say. Okay, दोनों के नाम शायद सेम ही है जैसे कि हमने देखा ट्रोपोनिन. Okay, एक केस में अल्फा है, सेकंड में बीटा है. Right? So like this. In the first case 1 and 2 is written and in the second case 1 and 3 is written. So this is what alternative exons or alternative splicing. Got it? Okay, so again this is the example alternative splicing of SV40 T antigen. Okay, अभी इसका DNA region क्या है? Exon, this is a 5 prime split site. Stop codon है यहाँ पे, stop codon. So this is the stop codon. Okay. Again here is the 5 prime split site and 3 prime split site. Okay. यहाँ पे we can see these are written in small alphabet and this is the capital alphabets. Right? So why so? See, यहाँ पे statement है splicing of the SV40 T antigen RNA. Both forms are typically produced and both proteins are made upon infection. मतलब infection के बाद दोनों proteins बनते हैं. The small t antigen means this one. In the first case, this is a small t antigen. क्या हो रहा है? Is encoded by the longer of the two mRNAs. Okay, longer of the two mRNAs. And that message contains an in-frame stop codon. Okay, this is very important. Upstream of exon 2. 5 prime SSRST refers to 5 prime split site. मतलब split site है वो. ये वाला. This is a 5 prime split site used to generate the large tmRNA. तो 5 prime split site जो small में लिखा है, that is the 5 prime split site used for small antigen, right? And 3 prime SST is the 3 prime split site used in generating both mRNAs. Okay, now I will explain. See, in the first case, यहाँ पे small t antigen है. We can see from the picture that this is small one and this is a large protein. Okay, I will write here. This is a small one and this is a large, right? Large है. Okay, यहाँ पे क्या है? This is a exon 1, this is a exon 2. But in this case, ये stop codon है, stop codon. So this stop codon is very important. Okay, we can see here, यहाँ पे stop codon है. So in the first case, इतने ही codon तक protein synthesis होगी. Okay, because this is a stop codon, right? Stop codon means translation end. Okay, so यहाँ तक stop codon तक के proteins बनेंगे in the case 1. इसीलिए हम इसे बोल रहे हैं small, okay, which is a T antigen. In the second case, what is happening? Stop codon नहीं, यहाँ पे retain नहीं हो रहा है stop codon. Just exon 1, exon 2 retain. यहाँ पे stop codon है ही नहीं. So protein synthesis continues. Thus, this protein is a large protein. But it's easy. In the first case, stop codon retained hai. In the second case, there is no stop codon. Just exons are joined and the protein length is large. And here stop codon की वजह से protein length is small. Got it? You have to remember that much only. Okay, so this is the next example of alternative splicing in the case of sex determination of Drosophila. Okay, see we can see here a cascade of alternative splicing events to determine the sex of fly, right? Okay, so यहाँ पे क्या हो रहा है? Just from a picture, what we can understand is that in the female, this is the female, right? And this is a male. Very simple. Just go through the male female pathway and compare it with the male pathway. Okay, दोनों का separate कैसा study कर सकते हैं? So this is a trick actually. First see the first pathway. Okay, यहाँ पे क्या हो रहा है? XSL protein. See, XSL protein is getting synthesized. Okay, by the process of transcription, translation, right? And this SSL protein is basically a regulator. Okay, we can see here splicing a regulator. See, this is a regulated 3' splice site. Okay, in the male case, there is no functional protein. SXL protein, sex lethal. Okay, sex lethal gene and sex lethal protein known as. So in the male, the protein is not synthesized. Female में ये protein है first protein which is regulating the 3' splice cell. Okay, again the next protein tra protein is synthesized. Right, tra protein. So this is a dimer, homodimer, tetramer formation taking place with the help of tra2. Okay, so ये structure अभी यहाँ पे आके bind होगा. Okay, see this is again a regulated structures. We can see a regulated 3' splice site in the tra gene and DSX gene. Tra is transformer. Okay, transformer and DSX is the double sex. Okay, I will write here. SXL is sex lethal. Transformer and double sex. Okay, so यहाँ पे क्या है? Regulated 3' splice site है. जब प्रोटीन बनेगा in the case of females, वहाँ पे regulation दिखा रहा है. And we can see here the structure is double sex protein in female is small. Okay, we can see here small hai, and in the case of male, this is big one. Okay, what it does, repress female development. Okay, repress female genes, and thus male development is done. And in this case, in the case of females, male genes are repressed, activates female genes, and thus female development takes place. Okay, statement what is written? ओके, सो यहाँ पे इंपॉर्टेंट है एक्स सेक्स लीथल प्रोटीन प्रोड्यूस हो रहे हैं इन फ्लाई दैट विल डेवलप इनटू मेल फीमेल्स. ओके, मेल्स में नहीं है, but those that will not develop into, okay, not present in males. The presence of that protein is maintained by auto-regulation or splicing of its own message, right? The SXL जो सेक्स लीथल जीन है वो खुद ही रेगुलेशन शो कर रहा है, right? In the absence of this regulation, no functional protein is produced. Okay, in the case of males, sex lethal also controls splicing of the TRA gene, producing functional TRA protein in females. Splicing regulator है TRA, SXL gene. TRA is itself a splicing regulator. See, TRA is also a regulator. It acts on pre-mRNA from the double sex gene. Okay, double sex gene के pre-mRNA पर ये act करेगा. When the double sex mRNA is spliced in response to TRA protein, a version of double sex protein is produced in females with a stretch of 30. This is very important. 30 amino acids at its C terminal. Okay, C terminal पर highlight किए. See this region. इतने amino acids है 30, and that distinguishes it. Okay, so this is a distinguishing factor from the form of protein produced in the absence of TRA regulator. Okay, मतलब यहाँ पे ये जो रीजन बन रहा है विच इज यूनिक, हाँ, प्रेजेंस ऑफ ट्रा प्रोटीन की वजह से, राइट? हियर देयर इज नो ट्रा प्रोटीन, so the female form of double sex activate gene regulate required for female development and represses those for male development. The male form, which has a, so this is having 150 amino acids at the C carboxy terminal end. Okay, we know that repress gene that the female development. Okay, SXL protein acts as a splicing repressor. See, splicing repressor by binding to the pyrimidine tract. Okay, कहाँ पे bind हो रहा है? This is very important point. At the 3' splicing site, the TRA protein in contrast acts as a splicing activator. It binds to an enhancer. Okay, इसके कारण ही मैंने यहाँ पे theory written की है, because there are some important words which are highlighting. I will underline again those important words. 30 amino acids in the females है, and in the male case 150 amino acids. ये important है, right? As well as the SXL protein acts as a splicing repressor. ये important है. कहाँ पे bind हो रहा है? pyrimidine tract पर and the 3' splicing site. This is important. Okay, फिर यहाँ पे TRA protein acts as a splicing regulator. ये भी important है. It binds to an enhancer sequence in one of the exon of the double sex RNA. SXL splicing repressor है. And TRA is a splicing activator. This binds to enhancer and this binds to polypyrimidine tract. Got it? Just highlighting points. Okay, here there is 30 amino acids and here 150, which is distinguishing factor. Got it? Okay, now this is the last example. Here you can see FOXP1 gene. So this is 17, 18A, 18B and 19. Okay, exons है 17, 18A, 18B and 19. Now in the differentiated cells में 17 है, 18B है and 19 है. Okay, 17, 18B and 19 in this case. Okay, यहाँ पे डिफरेंशिएशन होगा, सेल डिफरेंशिएशन, सेल डिफरेंशिएशन। Okay, we know this is a stem cell, right? Pluripotency, but pluripotency in the stem cells, stem cell में क्या है? Which genes are activated? 17, 18B and 19. Sorry, my mistake. Here it is 18A. In the differentiation case, here 18A is there, 18A, not 18B. Okay, 18A. And in stem cells, there is a 17. I will show here with pink. Then 18B and 19. So this is necessary for pluripotency. Okay, and no differentiation. Okay, so I will sum it up. This is also very important to slide actually. 17, 18A, 18B, 19. Okay, this is a FOX1, FOX P1. Right? In the first case, 17, 18B and 19 in stem cells. Okay, इसे हम FOX P1, FOX P1 ES बोलते हैं. Right, which is important for regeneration, regenerative capacity है उसमें. And the second type 17, 18A and 19, this is differentiation, differentiated cells. Okay, this is FOX P1 ही बोलेंगे. Got it? So this is in differentiation. Okay, differentiation. Got it? This will give rise to pluripotency. Pluripotency, right? So one more case to note here is that in this case, pluripotency in the stem cells, which genes are activated? कौन से genes activate होते हैं? OCT4. Okay, October we can remember OCT4 and NANOG. These are the two genes activated. You all must be aware of this word IPS, induced pluripotent stem cells. Okay, induced pluripotent stem cell means what? These are used in stem cell therapy. Very important part. Okay, मतलब इस genes को we can activate in the laboratory by using this alternative splicing method. Okay, so this is a FOX P1 gene. इससे two products मिलेंगे in the stem cell. अगर हमें activate करना है pluripotency, then FOX P1 ES, which is having 17, 18B and 19. Okay, we can see here. And if differentiation करना है, so FOX P1. Okay, in the differentiated gene. Okay, no pluripotency. Pluripotency is inhibited and differentiation is stimulated. Okay, got it? So these are the examples of alternative spacing. Let's summarize once again because बहुत कुछ देखा हमने. Firstly, we saw the introduction. Two different types of RNAs are produced, which will code for different proteins. Okay, here is the example: alpha-troponin, beta-troponin. We can see different exons are present. Then different types of splicing we saw. Okay, in which exon skipping comes, then exon extended, intron retained, alternative splicing, normal splicing. Okay, and then this in here we saw SV40 T antigen. This is seen in the virus. Okay, upon infection में हमें large protein कब मिलेगी, small protein कब मिलेगी, right? Then in the next case we saw Drosophila sex determination. Okay, female में protein factors बनेंगे, SXL, transformer and DSX. Okay, which are the regulators of splicing? एक negative regulator है, एक positive regulator है, right? And in the last case, we saw stem cells and differentiated cells, how we induce the pluripotency, how we can induce. Okay, so this is an example of regeneration, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Subscribe to the channel, guys, and stay tuned for the upcoming videos. Thank you.